లైవ్కి వెళ్ళింది కదా, ఇప్పుడు క్లియర్ చేయండి సార్ అందరికి.
మీది వెళ్ళిందా? ఇప్పుడు వెళ్ళింది. నాది వెళ్తుందమ్మా బాబరాజు. ఓకే. లైవ్ ఆప్షన్. Yes, sir. This one is live streaming right now. Kumar sir,
okay. Once it is done, I will share the screen.
Yes, sir. Sure, sir.
సరే మ్యూట్ చేయండి సార్ మీది. సార్ మ్యూట్ చేయండి సార్. మ్యూట్లోనే ఉన్నారు. ఒకసారి నేను చెప్పానండి, ఆయన్ని ఇంట్రడక్షన్ అన్మ్యూట్ చేసుకుని ఇచ్చామనండి. కుమార్ సార్ వచ్చారు, నేను ప్లే చేసుకుంటాను. ఆయన ఇంట్రడక్షన్ ఇచ్చామన్నాడు, స్టార్ట్ చేయమన్నాడు.
Okay. Good morning to one and all, and welcome to the day 6 and the last day of the FDP on nanotechnology in healthcare. Now it is my privilege to introduce our director sir to the gathering, who is going to give the welcome address. Dr. Kumar V.S. Nemmani, director since April 2018. He holds his B.Pharm and M.Pharm degree from Andhra University, Visakhapatnam. He has completed PhD degree from Naipur Mahali and subsequently postdoctoral fellowship from McGill University, Montreal, Canada. Dr. Kumar V.S. Nemmani has associated with 3 leading research centers in India. He has over 14 years of industrial research expertise and managerial experience. Now I request our director sir to address the gathering.
Hello everyone, and welcome to day 6. Today is the last day of this webinar series on nanotechnology in healthcare. I'm sure that all of you enjoyed the last 5 sessions, and the same will be true for sure that today's speaker will also engage you well with this, with his eminent expertise and work that he has done. He has a lot in store for sure. And he'll be enlightening all of us on role of nanotechnology, especially in the fields of, I guess, metabolic disorders and cancers. He has many projects, especially on metabolic disorders and nanotechnology in cancer. So on this note, on behalf of the management of Sri Vishnu Educational Society, our chairman sir, Sri K.V. Vishnu Raju Garu, our vice chairman, Sri Ravichandran Rajgopal Garu, and our principal, Dr. K. Prasad Garu, and all the staff of SVCP. Welcome you and welcome all the delegates. And thank you for taking your time and being part of this webinar series, this webinar pandemic. So there are many webinars happening at the same time, even though you all selected this seminar and being part of this for the past 5 days tells of the importance and the impact of nanotechnology in healthcare and the quality of the speakers that are present for this series. So at this juncture, I must thank the organizing secretary, Dr. Ashok, and the program coordinator, Dr. Ramchandra Rao Garu, for requesting and gathering the eminent speakers from the leading research institute from India and from the industry. So at this point, I'd like to say a few words about Dr. Praveen Krishnamurthy Garu. Thank you, sir, for accepting our invitation and being part of this webinar series.
My pleasure,
Being a fellow pharmacologist, I am very proud to listen to your talk and you being from the JSS, the premier institute in India, one of the premier institute in India, participating and enlightening all the participants is a really welcome note. So I must thank you for accepting and showing all your the work that is in store. So I'm really excited, looking forward for your talk. So at this juncture, I also thank just joined this webinar series. I'm proud to announce that from Monday onwards, we are organizing an international webinar series on drug discovery and development, and most of the speakers are from the extensive industry experience and from the leading institutes. So the way, the enthusiasm that you all shown for this webinar series, nanotechnology in healthcare, the same will be extended to drug discovery and development, which is coming up the next week. The following week will be on the pharmacy practice. So I'm sure you all participate in that too, or recommend to your friends who are the most suitable for this. So your feedback is welcome, and your honest feedback will help us refine our webinar series and the topic or the theme that we select to make this event more useful, meaningful, and fruitful to all of you in your future careers. So once again, I thank Dr. Praveen sir for accepting this invitation and welcome all of you for this wonderful talk. So forward to Ashok.
Yeah, it is my privilege to introduce the profile of today's guest speaker to the gathering. Dr. Praveen TK had done his B.Pharm from Kempegowda Institute of Medical Sciences, Bangalore, and his master's also from Kempegowda Institute of Medical Sciences, Bangalore. He did his PhD from JSS College of Pharmacy, Ooty. Later, he rendered his services at Himalaya Drug Company for a duration of 6 months. Later, he joined as a lecturer at Sri KV College of Pharmacy, Chikballapur for a duration of 11 months. Later again, he joined with Himalayan Drug Company in the Department of Pharmacology and served there for a duration of 2 years and 7 months. Later, he joined with as a lecturer as JSS College of Pharmacy, Ooty for a duration of 3 years and 3 months. Then promoted to assistant professor at JSS College of Pharmacy, Ooty. After working there for a duration of 6 years, 3 months, now he is currently working as a professor and head Department of Pharmacology at JSS College of the Pharmacy. Added to his credit, he published more than 555 publications in various national and international journals. He had conducted 45 research projects. He had one Indian patent, guided more than 16 PhD students. Today, he is going to deliver a topic on the preclinical assessment of nano-pharmaceutical toxicity. Now, I welcome Praveen sir to take over the session. Thank you, sir.
Thank you. Thank you so much. Can I share the, can you please enable the sharing option?
Yes, sir. You can share your screen, sir.
Okay. Yes. Still, it is not activated, sir. Can you please check it?
Now you can share, sir. It is
now. Yeah. Now I can share. Thank you. Okay. Good morning to all of you. So I thank Sri Vishnu College of Pharmacy for inviting me to be a speaker of this wonderful program. So I would like to also place it on record and thank the senior professor and director, Dr. Kumar, and also Dr. Ashok, who has taken a lead to invite me here. So in this topic, although I'm not a typical, nanotechnologist or somebody, I am a, jack of all, master of none. I do many things, but this is one of my interests. So where I have some projects, so therefore I have been forced to work in this area. So I would like to share some basic aspects of nanopharmaceuticals and also the regulatory point of view, how the current regulation deals with the clinical safety assessment of these nanopharmaceutical products. So before I would like to go into the whole thing about the toxicology aspects, I would like to talk about the nanoscience and technology, understand the little bit basics about this science. So nanoscience basically deals with the nanomaterials of or materials of the particle size range of 1 to 100 nanometers. Whereas nanotechnology is a science which deals with design, production, characterization, and applications of such nanomaterials by controlling their shape and size in the nanoscale range. So let us briefly understand what is the nanoscale range is all about. If you take example hydrogen atom, it is approximately of 0.1 nanometer size, and glucose molecule has 1 nanometer size, an antibody has approximately 10 nanometers size, and a virus is having 100 nanometer size, diameter of a virus, and then the bacteria is of 1000 nanometer size, and the bulk powders what we use for regular formulations is also the particle size of a bulk powder normally in 1000 nanometers or in micrometers we can say, and then an average human cell has a size of 1 lakh nanometers, and a dot what we keep on a paper is of 10 lakh nanometer size. So we can appreciate, so from an atom to a dot, how the size difference occurs, and nanoparticles basically we deal, by definition we say particles having 1 nanometers to 100 nanometers. However, when it comes to nanopharmaceuticals, we are not just dealing with particles or only with 1 to 100 nanometers. We go up to 200 nanometers, which I will be telling you the reasons why later on maybe in the discussion. So coming to why we use nanotechnology or why we use nanoparticles. Let us understand this nanotechnology concept with the example of a comparison, maybe a comparison of bulk material with the nanoparticles. So here you take the example of gold. You can see the bulk gold looks yellow. The solid gold bricks look yellow, and the powdered gold in the form of a bulk powder like micro size gold looks little reddish. And when you reduce this gold to a nano size, what happens? You will find different color for different size of nanoparticles. So there, depending on the nanoparticle size in the nano range, depending on the size of the particles in that nano range, different properties are exhibited by this gold nanoparticles. That means at a smaller size it will look reddish, and little bigger size at the nano scale it looks purplish. So what does that indicate? So in general, what is understood is that properties of a material such as physicochemical properties, biological, mechanical, optical, electronic, and other properties change as their size approaches the nano scale. We all appreciate that when the gold is reduced to nanoparticle size, its properties changes. Okay, there are so many properties which change. For example, in this case, we are just seeing the color. So in addition, a bulk material will have a constant physical properties regardless of size in that particular range. For example, you take micro gold. This picture you can see there are some gold, these micro gold particles are small, some are lumps which are big, but all these particles, irrespective of their size differences or size distribution, they have a similar physicochemical properties. This you all should understand. In general, we say bulk material within the size distribution of their particles possess same physicochemical properties. But nanoparticles differ with the bulk materials in this aspect. That means you can see in this nano gold, the nanoscale, their properties change depending on their size in the nanoscale. If in the nanoscale, if they, for example, it is 1 nanometer, it has a different property. If it is 10 nanometer, it has a different property. If it is 100 nanometer, it has a different physicochemical properties. So that is how a nanomaterial differs with the bulk material. In case of a bulk material, within the size distribution range, all the particles will have the same physicochemical properties, whereas nanomaterials, when the size changes or shape changes or the surface properties changes within the nanoscale, also results in change in the physicochemical properties. This you all should remember to understand the basics of nanotechnology. So, in addition, nanomaterial characteristics are not only determined by the chemical composition, but also by their morphological properties, such as size, shape, and surface properties. We will appreciate this concept when we understand with some examples later in the presentation. So what here I would like to say is that nanomaterial properties are not just dependent on their intrinsic constituent properties. It also depends on their surface properties like shape and size as well. And the question now arises, why these materials possess different physicochemical properties at a nanoscale range? This is because nanomaterials possess high surface area over volume ratio, or in other words, they have a high surface to volume ratio, and also high percentage of atoms are present at the surface in case of narrow material when compared to the total number of atoms. If you take this bulk material, number of atoms present on the surface is relatively very minimal when compared to the total number of atoms present in that bulk material. Whereas when you reduce this bulk material to a small nanoscale, what happens? So these materials come closer to a molecular size or subatomic scale size, where the number of atoms present on the surface will significantly increase compared to the total number of atoms. So for example, if you take a rough example, if I have to give, if you take a bulk material of micro scale, what happens? The number of atoms may be present on the surface may be 100 out of lakh atoms, which are total atoms. When it comes to a nanoscale, the number of atoms present may be in lakhs when compared to the total number of 10 lakhs. So therefore, what happens? The number of atoms increases at the surface. If you take one of the properties of such material, reactivity, what happens? In case of nanoscale, there will be more number of atoms are available for reaction. Therefore, nanomaterials will be more reactive than the bulk materials of micro scale size. And also, another important aspect of nanomaterial characterization is that, so nanomaterials, okay, they are of the range of, by definition, 1 to 100 nanometers. They come closer to a molecular size or an atom size. So therefore, they follow quantum properties. So their properties or their physicochemical properties cannot be just explained by using classical mechanics. So classical mechanics is normally used for defining the physicochemical properties or the motion of a material for large particles. Okay, for example, you see in ISRO launches a rocket, you see the path of the rocket is predicted by using the classical mechanics principle, because the rocket is of a larger size and that is predicted by classical mechanics. When the size of the particle material decreases, when it comes to atom or a molecule or subatomic particles like electrons, you cannot define their movement or physicochemical properties just by using the classical physics. Instead, you need to adopt the principles of quantum physics. So here, the meaning of saying the quantum size effects will take into effect means the nanoparticles start behaving like atoms or molecules. Start behaving like atoms and molecules. So this is what the basics I would like to give you regarding the nanotechnology. And because of this small size effect, high volume to surface ratio or high number of atoms which are present in the surface provides different physicochemical properties for the nanoparticles. which will make them useful in various fields. In fact, nanoparticles have played a very important role in determining the size of your mobile phones, what we are all using today, and even the computers, what we are using. Nanoparticles, nanotechnology has a significant role to play. In addition to electronics, nanotechnology has played a very important role in environmental sciences, in renewable energy, textiles, biomedical sciences, healthcare, food and agriculture, and even in industrial production. I'm not going into the details. You can later on see this, or you can look into the literature. So my focus today is on nanopharmaceuticals or nanomedicines. The application of nanotechnology in medicine field. There are huge number of benefits, okay, which nanotechnology can offer, especially in site-specific delivery of drugs to a particular tissue or to a particular site in a body. So before we get into that, let us understand what are nanopharmaceuticals. A nanopharmaceutical is defined as a pharmaceutical preparation containing nanomaterials intended for internal use or external application on human for the purpose of therapeutics, diagnostics, and health benefits. So what is the rationale for the development of nanopharmaceuticals? So the rationale is nanoformulations offer unique physical, chemical properties at cellular, atomic, and molecular levels not usually seen with the bulk formulations. Once again, this property may be due to their high surface to volume ratio and high number of atoms present on the surface of these nanoparticles. Another important application of nanopharmaceuticals would be to provide flexible options for drug delivery, developing and designing novel drug delivery systems, which can provide a lot of advantages, such as ability to overcome biological barriers. For example, if you want to take niclosamide, is one of the anti-helminthic drug. It does not have a good bioavailability, but it shows in the in vitro experiments the anti-cancer properties when it has got an ability to destroy cancer stem cells. So one of my scholars has worked on this area where we try to develop this niclosamide as a nano formulation to repurpose this drug, where we could increase its bioavailability by converting this into a nano formulation. So therefore, we can nano formulations by default can help cross biological membranes. For example, blood-brain barrier. Many drugs do not make it to the clinical trials or do not succeed in the clinical trials because many of them do not have reach any appropriate concentration in the brain to treat various neurological disorders. Such medications or such potential compounds can still be repurposed by using nanotechnology, because nanotechnology can provide options for designing a drug delivery system which can overcome these biological barriers. And ability to deliver hydrophilic or hydrophobic molecules. Nano carriers can be used, to carry drugs which are highly hydrophilic or highly hydrophobic. Otherwise, these molecules, in their bulk form, will not have sufficient bioavailability. When you convert them to nano formulations, so there are chances that these medications can still be repurposed. And another important aspect, especially for developing nanopharmaceuticals, would be ability to selectively deliver drug cargo to specific sites in the body. Especially in cancer targeting, nano formulations have been extensively used for deliver drugs specifically to the tumor sites to avoid off-target side effects. So based on this rationale, the applications of nanopharmaceuticals, some of the applications I have listed here, these include active or passive targeting. Nanopharmaceuticals can be used for active or passive targeting, especially they are extensively researched in the area of cancer treatment, and sustained or controlled release effects. So nanopharmaceuticals not only carry cross the blood-brain barriers or the biological barriers, they can also control the release of these medications, and thereby can improve the patient compliance and reduce the cost of therapy as well. And then improve solubility of many medications, because when you reduce the size, the surface area increases, and therefore the solubility of a substance can increase. And also they significantly increase the bioavailability by various mechanisms. One is obviously by increasing the solubility, and also by protecting the drug from degradation in the circulation. They can increase the circulation time of a drug, and thereby they can indirectly increase the bioavailability, and also by carrying the drug molecule in a larger quantity at the receptor site. And another important application of nanotechnology in the field of pharmaceuticals or in the field of medicine is the stimuli-sensitive release of drugs. So what I mean here is that nanoparticles, especially polymeric nanoparticles, can be designed such that they release the rate of release of the drug can be enhanced by a particular environment. For example, you take tumors that have higher temperature or lower pH. So you can exploit these differences in the tumor environment and the normal physiology to selectively release the medication in the tumor environment. That means here the stimuli would be pH or the temperature. So once the higher temperature is there, the polymer will try to increase the rate of release of drug in the tumor site, thereby providing a more selective delivery of drug to the tumor site. And obviously, by active or passive targeting, you are going to achieve site-specific delivery of the drugs, and as a result, you are going to reduce the off-target side effects. Therefore, nanotechnology can help in reducing the toxicity as well. Then therapeutic efficacy is obviously going to be increased when you deliver theoretically when you deliver a drug through nano carriers, so where they have the ability to deliver the drug more efficiently to the target sites. Therefore, therapeutic efficacy indirectly increases, and they can also be used as a precision medicine. And in addition, many of these nanoparticles possess diagnostic. That means at nanoscale, some of these materials can possess optical properties. As a result, they can be used for diagnosis in addition to therapeutic benefits. The new term coined based on these two concepts is the prognostic applications. Nanotechnology has been extensively researched for their prognostic application, that is both diagnosis as well as the therapeutic applications. Let me give you some of the nano formulations which are there in the market and their applications advantages as well. So most commonly available nano formulations as on today in the market belong to the following three categories, that is nanocrystals, polymeric nanoparticles, and liposomes. So these are the most commonly available nano formulations in the current market. So nanocrystals are crystalline particles produced by different methods such as milling, high pressure homogenization, and by precipitation process. Advantages of these nanocrystals is obviously they improve the dissolution rate and also saturation solubility, and hence increase the bioavailability. They have been applied in various disease conditions like cancer, atherosclerosis, and all. Polymeric nanoparticles, various polymers can be used to incorporate as a carrier system for various drugs, and they have various advantages like they can be administered by oral as well as by parenteral routes, and they have tunable characteristics like you can tune them for stimuli sensitive release, and they can carry multiple drugs as well, and they can improve the stability of the drug cargo, they can protect the drug from degradation, and also they have a good cell penetration capabilities. Similarly, liposomes, where drug is incorporated into the lipid membrane, in a lipid cavity or bilayer, in which both water soluble as well as the lipid soluble drugs can be incorporated, and liposomes can carry water soluble drugs in their water core and lipid soluble drugs in their peripheral lipid bilayer, and they can be used for passive targeting of the drugs, and they are highly efficient cargo for delivery of drugs to the tumor site and many other sites in the body, and they can also reduce the cargo toxicity. Let me give this a brief about some of this, explain the advantages of these nanocrystals, these products which are there in the market. For example, you take mint, which is the active ingredient is aproprint, which is an example for nanocrystal. So this aproprint is a neurokinin 1 receptor antagonist, which is used as an anti-emetic agent to control the chemotherapy-induced emesis. So this aproprint has been prepared as a nanocrystal to improve its bioavailability and also solubility. And then Ostim is another product, which is calcium hydroxyapatite. And another product is the Vitos, which is a beta tricalcium phosphate. So these two, once again, are used in bone grafting, in dental surgery, as well as in orthopedic surgeries. And the other drug is the rapamune, which is ripamycin. We all know that ripamycin is a immunosuppressant drug, which is used as an immunosuppressant, which is approved by US FDA in 2010. And other drug is the Ritalin, which is methyl phenidate, which is used in attention deficiency syndrome. And the last drug is in this food category of nanocrystals is the core, which is also called as fenofibrate. The active ingredient is the fenofibrate. Fenofibrate, we all know that it's a PPR alpha agonist, which is used as an anti-hyperlipidemic agent. So let me explain with the help of this fenofibrate how this nanocrystal form of fenofibrate has helped in improving the bioavailability. Fenofibrate in its bulk form, actually it is highly insoluble in water. It has got a very poor water solubility. As a result, it has very little absorption from the gastrointestinal tract, because you all know that a drug, before getting absorbed, it should be soluble in the gastrointestinal tract. So as a result of its poor solubility, it used to have a very low bioavailability in its bulk form. So conversion of this fenofibrate to nanocrystals has significantly improved its solubility and also significantly improved its bioavailability. As a result, unlike previously, fenofibrate was given along with food so as to facilitate its absorption with the help of bile juice or the bile salts. But now fenofibrate can be given before food or even after food, and there is no need to fenofibrate to be given along with the food for its absorption. So this is how nanocrystals will help in improving the solubility and bioavailability of some of the drugs. Some of the polymeric nanoparticles which are there in the market. So you have Cimzia, which is a citrulizumab. It is a pegylated citrulizumab, and this is a monoclonal antibody. We all know that it is directed against the TNF alpha receptors, and it is used for controlling the inflammation in autoimmune disorders like rheumatoid arthritis. The other drug is the Rajan, which is pegylated adenosine deaminase. So excess adenosine accumulation is implicated in severe combined immunodeficiency disorders, which is found to affect the lymphocytes function. As a result, there is a immunosuppression. So this adenosine deaminase enzyme is used in nano-formulations to improve its efficacy. We'll understand how it will improve by formulating these drugs in the nano-formulation, how they will improve their bioavailability. And then another drug is Neulasta. Neulasta is once again a pegylated filgrastim. Filgrastim, we all know it is a colony-stimulating factor which stimulates the neutrophils, which is normally given in HIV patients or immunocompromised patients. Similarly, asparginase, that is oncospor, which is pegylated L-asparginase, which is an enzyme which is converted into a nanoform and then which is delivered, which is normally given in the chemotherapy of cancer. And then Pegasus is another drug which is pegylated alpha-2 interferon, which is again used in various immune-related disorders like in hepatitis, which is used to improve the immunity. And then last one is the Macugen, which is pegylated, pacatinib sodium, which is used for controlling the macular degeneration or treating the macular degeneration. So if you look into all these medications, there are some of proteinaceous medications, and except maybe pacatinib. So they all have immunogenic properties. If you give them directly, for example, you take Cimzia, which is the monoclonal antibody, that is citrulizumab, which has because it's a protein, it is highly immunogenic. It means if you give it directly, what happens? Our immune system will try to destroy the drug, and thereby its efficacy decreases, and it is having it causes an immune reaction. In addition to that, it does not serve its purpose. So therefore, what
Raju.
Sir, Praveen sir. Sir, Praveen sir. Sharing what I can.
It looks like there is a power failure at his end.
Dear participants, sorry for the inconvenience. There is some internet connecting issue at the guest speaker's venue. So please, inconvenience is kindly regretted. Thank you all.
హలో నరసింహారెడ్డి గారు
పవర్ ఫెయిల్యూర్.
హలో ఎవరు?
నేను మీ పాత మాస్టర్ని భీమవరం నుంచి మాట్లాడుతున్నాం.
నమస్కారం దయా.
ఏం మంచిది, రిలాక్స్డ్ గా కూర్చున్నావు.
హలో హలో టీచర్
Praveen sir, hope you are back. Yeah, thank you, sir.
I'm there. Thank you.
We can able to see your screen, but keep in F5, sir. Full screen mode.
Yeah, I'm doing that.
Thank you, sir.
Okay. I'm extremely sorry for the interruption. So I was talking about the application of polymeric nanoparticles. I was just telling that polymeric nanoparticles can significantly increase the circulation of time of the drugs, and also they can protect the proteinaceous drugs from degradation in the serum or in the another very important class of nanopharmaceuticals which are there in the market. There are so many anti-cancer drugs like doxorubicin, vincristine, citalopram, and then the antifungal drug amphotericin B have been successfully formulated as liposomes, and they are currently available in the market. And liposomes, like polymeric nanoparticles, can also protect the drug from degradation in the circulation. Therefore, they can also improve the circulation time, and as a result, they can improve their bioavailability. And also, liposomes being more lipophilic, they can carry hydrophilic drugs which are otherwise cannot cross the biological membranes. So thereby, they can improve their bioavailability. And liposomes can have enhanced permeation and retention effect, especially in the treatment of cancers, and thereby passively delivering the drug more to the cancerous tissue than to the off-target sites, and thereby reducing the off-target side effects. These are some of the advantages of liposomal formulations. So these are some of the other nanocarriers which can be developed, and some of these formulations are also maybe there in the market. So some of these include magnetic nanoparticles, which you can use deliberately, use the external magnetic field to deliberately concentrate a drug in a particular site in a body, and then micelles, dendrimers, carbon nanotubes, gold nanoparticles, quantum dots. Quantum dots have a lot of application in the biological optical detection and then for diagnostic purposes. So coming to the global market share of the nanopharmaceuticals, so in 2016, it was estimated that 111 billion US dollar was the total market share of this nano pharmaceuticals. And in the year 2025, it is expected to reach 50 billion US dollars. And this is the global market share of nanopharmaceuticals. And these products are expected to grow at a rate of 11.2% year on year. And the challenges faced by nanopharmaceuticals. If you see the conventional formulations and compare that with the nanopharmaceuticals, their number are very less. And we can ask a question why they are very few in numbers. It's not easy to develop a nanopharmaceuticals, unlike a bulk formulations. They are not that easy to develop. Let us understand what are the challenges faced by these nanopharmaceuticals. So one of the challenges is also the preclinical development of these nanopharmaceuticals. So the first one is as you all know, that the nanomaterials possess different physical, chemical, and biological properties when compared to the bulk counterparts. And further, when either you directly convert a API, active pharmaceutical ingredient, into a nanopharmaceutical, or you try to load a API into a nanocarrier, so these, as a result of their unique physicochemical properties, they show different biological properties like different pharmacokinetic, pharmacodynamic, and toxicological properties. So in case of an API, this is not a problem. We know that we can predict the biological properties like pharmacokinetic, pharmacodynamic, and toxicological properties for an API very consistently, because from different size ranges of an API in micro scale, they do possess similar biological properties, what I just listed out. However, in case of nanoparticles, it is not the case. Please go back and remember this picture and compare the micro gold with the nano gold. So what happens? The variation in the pharmacokinetics, pharmacodynamics, and toxicological properties do occur within the nanoscale range. Suppose you prepare one batch of a formulation in one particular nanoparticle size, it will have a different biological properties. And if you next batch, there is a little variation, then it will have a unique different biological properties. As a result, this is one of the biggest challenge I believe which a nano formulation faces in the to becoming a real formulation compete with the other formulations. Another important challenge would be nano formulations are complex in nature. That means if you see a regular tablet formulation, what we do is we take the API and then we add some excipients and we punch and the tablet is ready. And we do the disintegration dissolution test, some of the tests, and you can have a very good control over these quality parameters. However, nano formulations are complex and they involve many components and many steps of synthesis and preparation. For example, they may contain a prodrug, they may contain a active drug, or they may also have a tracking agent along with that, and they may have a targeting molecules attached to them, like monoclonal antibodies which are targeted against the particular protein in the body, and also they carry many surfactant and so many other components are carried by these nanoparticles. And as a default, their design itself is very complex because they are intended to achieve some extra things. So in that context, what happens? The manufacturing and then quality assessment becomes very complex. So this results in a complexity in the evaluation of their quality, safety, and efficacy. So we will understand these aspects little in details in the next few slides. Let us understand some of the formulation challenges. So nanopharmaceuticals are normally prepared by two methods, top-down methods or by bottom-up method. Excuse me. In top-down method, what we do is, basically the bulk material is broken down to form nanoparticles. In case of bottom-up method, from a molecule, what happens? Molecules are clustered together to form nanoparticles. Some of the examples for top-down methods include high-pressure homogenization, per milling, thermal laser ablation are some of the examples for top-down methods which are employed for preparing the nanoparticles. And coming to the bottom-up method, you have precipitation method, vapor deposition method. Vapor deposition method can just be example for your vehicle exhaust where carbon which deposits, that itself is a nanoparticles, can be a very good example for this. And spare pyrolysis can be another example for preparation of this. Atom and molecule condensation are some of the other methods which are used for preparing these nanoparticles. And if you see the steps involved in the nanoparticle preparation, and you have size reduction, homogenization, centrifugation, sonication, and solvent evaporation, lyophilization, extrusion, and sterilization. There are so many steps which are involved in the preparation of these nanoparticle formulations. As a result, when multiple steps are involved, however, at an industrial scale, sophisticated equipment, and also well-trained technicians, you cannot see nanoparticles themselves can be toxic. They produce so many toxic effects at the manufacturing place. That means they can be an environmental hazard. Therefore, you need to have a specialized equipment which can protect the people who are involved in the creation of these nanoparticles. So that may add up to the cost. And as a result of this default intrinsic challenges, it's very difficult to control batch-to-batch variation when it comes to nanoparticle pharmaceuticals. That means what? When there is a batch-to-batch variation, there is a batch-to-batch variation in the pharmacokinetics, pharmacodynamic, as well as the toxicological profile of that nano formulation. So this is one of the major challenge, I believe, when it comes to nano pharmaceuticals. Let's take some of the examples. If you take, for example, particle size and distribution, how it can influence the biological activity as such. So particle size and distribution cannot be accurately controlled at large scale manufacturing. You cannot have a batch which say, for example, the mean particle size is 150 and the particle size distribution is around mean plus or minus 20. That means between 130 to 170, you cannot have that kind of a preparation in every batch. So how particle size influences the biological activity or preparation as a whole? For example, particles which are less than 30 nanometers are expected to be rapidly cleared from the kidneys. And particles which are greater than 200 nanometers are normally recognized by the immune system as antigens, and they are going to be cleared off by the reticular endothelial system, which removes the nanoparticles from the bloodstream and thereby reducing their bioavailability. So ideally, a nanoparticle should have a size range between above 30 and below 200 nanometers to show some biological properties. So when this is the case, if your manufacturing is not able to control this size and particle size and its distribution, then it becomes very difficult to gain the benefit of these nanoparticles. Therefore, to achieve the benefits of nanomedicine, size and distribution should be carefully controlled in the manufacturing. So this is how size can influence the bioavailability as well as the therapeutic and toxic efficacy of the nanoformulation. Then another example, another physicochemical parameter which can influence, which can be challenging to control, with the surface properties. Surface properties such as charge, functional groups, hydrophobicity on the surface or hydrophobic groups on the surface, or the polymer which is present on the surface and its conformation can significantly influence how these nanoparticles interact with the proteins as well as with the cells. And further, this can further influence its stability, its circulation time, as well as the clearance by the reticuloendothelial system, RES system, or immune clearance. So normally what happens whenever you give a nanoparticles are considered as antigens. They normally absorb proteins present in the serum. See this example. What happens, these proteins can be C3 opsins, or it can be like immunoglobulins which can absorb onto these nanoparticles, or even albumin and so many other proteins or lipoproteins can absorb onto these nanoparticles, and they, as a result of this absorption of these proteins, create a corona around these nanoparticles, and this will promote clearance by the macrophages. So this process is called opsonization. And further, if the nanoparticles have lipophilic groups, then they can directly taken up by these macrophages and therefore clear. So that means what? Nanoparticle bioavailability is significantly decreased if you don't modify their surface. Therefore, surface modification is normally carried out with the hydrophilic polymers, like most commonly used polymer for obtaining the stealth properties or creating the stealth properties for these nanoparticles is the polyethylene glycol, PEG. So pegylated nanoparticles. Remember in the previous examples I gave for the polymeric nanoparticles, I showed pegylated polymeric nanoparticles as an example. So pegylation, what it does, if you add a pegylated group of polyethylene glycol group on the surface of the nanoparticles, they will try to repel these proteins which are present in the bed, and thereby prevent corona formation or adsorption of the protein onto these nanoparticles, and thereby prevent their clearance by the macrophages, and thereby increasing their circulation time. So now the question here is that during a preparation of a nano formulation or a nanoparticles API, what happens? You need to accurately control the surface properties. It is not that easy. Right, about controlling, but also analyzing that accurately the surface properties is also not very easy to maintain a quality product. Therefore, identifying the appropriate analytical test fully characterize nanomedicine, whether physical, chemical, or biological, may be one of the more challenging aspects of nanomedicine development, both from a technical as well as a regulatory perspective. You cannot very accurately define the surface is like this, and you cannot maintain that surface properties in every batch. This is one of the another biggest challenge in case of nanopharmaceuticals. Coming to the pharmacological challenges, that is challenges in terms of efficacy. So when compared to normal bulk drugs, you take for example paracetamol. I put the structure of paracetamol here only. What we do is, so we measure the paracetamol concentration in the blood, and we define minimum effective concentration, that kinetic curve we create, and in that we define minimum effective concentration and therapeutic window and maximum tolerated dose or concentration, and all those things we define in the blood. So in case of bulk drug, easily, and they quickly diffuse across the biological. I'm sorry, I lost the connection again. Yeah. I'm reconnecting now.
Yes, sir. You can share your screen, sir.
Yeah, I'm just doing that. Is it visible now?
Yes, sir.
Okay, what I was telling is that in case of a bulk formulation, though the measuring the concentration of the drug in the bloodstream can also be indirectly taken as the concentration which is present at the receptor site. But same cannot be applied to a nano formulation, because the drug which is present inside a nano carrier, concentration which is measured in the blood may not correlate the receptor site how we correlated in terms of a bulk molecule, a bulk API. So because the further release of the this bulk API from the nanoparticle carrier is in turn dependent on the physicochemical properties of the nano carrier. Okay, such as its size, shape, and surface properties in turn determine how this drug material is released at the receptor site. In further, there are so many biological factors which may also increase the release of these nanoparticles and drug from the nanoparticles, for example, like lysosomal degradation, enzyme, and so many physiological pH, and all those things will determine how this drug molecule is going to be released at the receptor sites. So therefore, when it comes to nanopharmaceuticals, you cannot simply measure the blood concentration and correlate that concentration as the receptor site concentration. Is therefore more appropriate for a nano formulation to measure the concentration at the tissue sites rather than at the blood. And further, it would be more appropriate if you compare the pharmacokinetics of nanomedicine as a whole in addition to the bulk API, so that you can get a difference in the pharmacokinetics to make a better decisions. Then the safety challenges is another very important challenge which nanopharmaceuticals face. So unfortunately, physicochemical properties which give unique advantages as a drug delivery system to the nanopharmaceuticals are also responsible for the safety concerns or causing toxic effects in the body. For example, you take size, shape, and all these things, and their ability to interact with the proteins and their ability to interact with the receptors can also lead to their toxicity. One of the major cause for toxicity is their high surface to volume ratio and their high surface reactivity, because of more number of atoms present on their surface can account for generating free radicals. Major mechanisms of toxicity of nanoparticle formulations include association with the cell membrane. Many of the nanoparticles can associate with the cell membrane receptors. For example, you take here this diagram, the right side diagram I'm showing, nanoparticles can reported to interact with the epithelial growth factor receptors, as well as calcium channels, as well as integrin receptors. And by interacting with the epithelial growth factors, they are reported to promote apoptosis. And by interacting with the calcium channels, they are going to increase the calcium levels in the cell. You know that in excess intracellular calcium can be toxic, which can activate the apoptotic pathways and can cause a programmed cell death. And then many of these nanoformulations can release toxic metabolite, toxic materials inside after they get dissolved inside the biological fluids, and that can cause direct toxicity. And the major toxic mechanism would be oxidative stress. So many of the nanoparticles themselves can generate free radicals, and because of their high reactivity, in addition, they can interact with the proteins and can generate free radicals. For example, in case of lysosomal in the lysosomes, the presence of nanoparticles can increase the free radical generation in the lysosomes. These free radicals which are generated can cause various organelle damage, various membrane damages, and they can also cause direct toxicity to the genome and then result in various types of toxicities. So nanoparticles below 100 nanometers are reported to can enter easily cells, whereas the nanoparticle size below 40 nanometers can easily enter the nucleus. Therefore, their toxicities can be immense, and care should be taken when designing a nanopharmaceuticals. And another important thing you need to keep in your mind is that for those materials which are generally accepted as non-toxic in their bulk form, found to show some toxicity when the same material is converted into a nanoscale and administered. So this is one of the major concerns in terms of safety of nanopharmaceuticals. They can really become toxic at nanoscale. And also another important aspect is, since they are this nanoscale level, they can also undergo accumulation in the body. Unlike drugs which will undergo metabolism and they are easily cleared after some time, but nanoparticles can remain in the body accumulated for a longer period of time, which further can promote such kind of toxicities. These are some of the reported toxicities and organs which are reported to undergo, damage due to nanoparticle administration. In the brain is one of the organs which can result in various neurological disorders like Parkinson's disease, Alzheimer's disease. Lungs are reported to be damaged or because of the nanoparticles. Exposure like asthma, bronchitis, emphysema, cancer have been reported after exposure to nanoparticles. And then circulatory system, heart, all lymphatic system and gastrointestinal system. All these systems have shown some degree of toxicity when they are exposed to these nanoparticles or nanopharmaceuticals. So coming to the nanopharmaceutical preclinical safety evolution. So we need to be sure, before we all know that in the drug discovery process, from target identification, target validation, lead identification, lead validation, then we have a preclinical development where we do preclinical safety testing before we go to the clinical development. So in the drug discovery and development process, preclinical safety evolution plays a very integral role, wherein which we try to assess the safety of a medication by testing in preclinical subjects like in animals. So for nanopharmaceuticals, unlike regular medications, regular drugs, we don't have a uniform internationally accepted guidelines for nanopharmaceutical safety evolution. Obviously, the reason is we all know that nanopharmaceuticals are very unique and their properties change when they have a slight changes in their particle size or surface properties. As a result, it is very challenging to develop uniformly accepted guidelines to evaluate these products. As on date, there is consensus to evaluate these product by product basis and by considering their physical, chemical, and biological characteristics. So no regulator has come out with a uniform guidelines. They only given some suggestions with respect to how to go about assessing the safety of these products. So as on date, therefore, the strategies used for developing the conventional medical products have been applied for nanopharmaceuticals as well for assessing their safety, toxicity, and compatibility. That means the same guidelines are used, but extra care is taken wherever necessary, and regulators are giving approval based on the product by product, and there is no uniform guidelines available. And Department of Biotechnology, along with CDSCO, and then ICMR has come out in 2009 a recent guideline for evaluation of nanopharmaceuticals in India, and this guideline also will only give you the general, I will tell you to analyze this product based on the conventional guidelines available for the current medicinal product. However, they tell you to look into some of the special characteristics, like for looking at this characterization of their surface, particle size, and all those things has been given importance. I'll quickly go through this brief into this guideline before I get into the some of the studies, preclinical studies. So classification of nanopharmaceuticals as far as this regulatory guidelines, they can be classified based on their nature. For example, based on their degradability, they can be classified as biodegradable as well as non-biodegradable. That means this classification is given to classify the application of nanopharmaceutical into any of these categories. Biodegradable, as the name suggests, these are the nanoparticles which are easily biodegradable, like polymeric nanoparticles and containing many of the drugs. And then non-biodegradable, you have gold
Hello.
Can you see the slide, please?
Yes, sir, we can.
Sorry, once again. Okay. So as I told you in the beginning of the regulatory aspects of these nanopharmaceuticals, nanopharmaceuticals are evaluated by the regulators based on the product by product or the case by case approach. Although this approach has been employed, still there are some general guidelines or general data requirements which are proposed in this guidelines in general, where manufacturer wants to carry out the clinical trials or want to manufacture, who want to sell a nano formulation in India should provide all this data before getting the approval. Some of these I'll quickly go through because of the want of time. Some of these include justification for development of the nanopharmaceutical. You need to have a strong justification how your nanoformulation is going to be unique and advantageous over the bulk formulation. That has to be justified for applying for an approval. Then chemical and pharmaceutical information. This may include information on the ingredients, the API, as well as the nano carrier, and then physical characterization data of the nanopharmaceutical. Physical characterization data is very important in terms of characterizing their particle size and their surface and all those things. Therefore, this include drug loading capacity, shapes, surface, texture information, etc. Should be provided. And then encapsulation efficiency and so many other aspects has to be provided. And the other important aspect is the analytical data for analyzing the nanocarrier, API, and nanopharmaceutical should be given, like size determination by using the STEM and SEM method, and NMR spectra and FTIR, fluorescence spectra, Raman spectra, UV visible spectra. All this data should be given. Complete monograph specification for nanopharmaceutical should be given. And analytical method validations for nanopharmaceutical analysis should be shared. And stability studies of nanopharmaceuticals in various conditions should be available. And data on nanopharmaceutical formulation like dosage form, route of administration, composition, all should be provided. And in addition, comparative evaluation of inventor product or approved Indian product, if available, should be given. Unfortunately, this is very challenging to find. So one of the biggest regulatory challenge for nanopharmaceutical evolution is that there is no standard available for comparing. There is no standards available for comparing because of their variable, or very difficult to control manufacturing. And then animal pharmacology data is another very important aspect which need to be provided by the inventor, like specific pharmacological actions which are required for therapeutic benefits. General pharmacological actions also should be available, and essential follow-up and supplementary safety pharmacology studies on the vital organs such as lung, kidney, liver, all this should be available, and also obviously pharmacokinetic data should be available. And then animal toxicological data required include systemic toxicity studies like acute and repeated dose systemic toxicity studies, male fertility studies, and female reproduction and developmental toxicity studies, that is segment 1, segment 2, and segment 3 studies, and local toxicity studies, and then allergy studies and hypersensitivity studies, and genotoxicity and carcinogenicity studies are some of the studies which are required to be provided for the regulatory approval. Finally, human, after all these things studies, you need to also obviously provide clinical study data, so for further approval. These are all some of the general guidelines which is given in the regulatory regulations for analyzing the nanopharmaceuticals. So I will be just briefly explaining about some of the regulatory aspects in terms of animal pharmacology data and in terms of animal toxicology data, and what are the care and caution which need to be taken when analyzing the nanopharmaceutical in comparison to a bulk formulation. These are the same guidelines or requirements which is also holds good for the regular pharmaceutical drugs. In fact, the same is adopted for analyzing the nanopharmaceuticals. But however, nanopharmaceuticals may require certain additional information. So in case of animal pharmacological data, So to evaluate the efficacy of nanopharmaceutical, preclinical testing should generate data for enhanced therapeutic efficacy. So whenever there is a rationale, whenever you are using preparing a nanopharmaceutical, obviously you say that there is some advantages. You need to prove that by carrying out in the preclinical studies, for example, enhanced therapeutic efficacy when compared to the bulk counter that has to be established in animal studies. Possible accumulation of drug at the disease target site or enhanced overall bioavailability, circulation, and biodistribution has to be established for the nanopharmaceutical. And one should establish off-target effects of the nanopharmaceuticals. And disease site specific retention and metabolism of the drug data should be available. And data should be available to differentiate between the bioavailability of the drug and the concentration of the drug at the site of action wherever possible. And also data should be available to help evaluate how plasma, off-target tissue, and disease site pharmacokinetics is affected by repeated dosing. These are some of the points which one should keep in mind when evaluating a nanopharmaceutical or generating animal pharmacology data. When it comes to toxicology data development, to evaluate the safety of a nanopharmaceutical, preclinical testing should generate data for establishing the systemic exposure achieved in animals and its relationship to the dose level and time course of the toxicity study. In the sense, at the highest dose, what is the maximum blood levels of these nanopharmaceuticals or the tissue levels of this nanopharmaceutical is going to be achieved, has to be available whenever you are doing a toxicity studies. Toxicokinetics data is another very important data which you need to establish. What is the difference between pharmacokinetic data and a toxicokinetic data? Pharmacokinetic data is generated in normal doses. Normally, it is done at a therapeutic doses. When you do a toxicokinetic data is generated at high doses, normally at toxic doses. So, for example, this type of data is normally generated as an integral part of a preclinical toxicity studies. For example, you are using, in case of repeated dose toxicity, you are using 3 dose levels, say 100, 500, and 1000 mg per kg. Normally, 1000 mg per kg is considered as the highest dose, and at that dose, what happens? Saturation kinetics will take into effect. So, in such a situation, the pharmacokinetics will not be the same. So, there you try to establish how the movement of the drug, the body from one compartment to another compartment changes. To make it very simple, for example, imagine when you give suppose one of the drug, for example, you imagine a drug is transported across a membrane by a facilitated diffusion like glucose. So when you give the drug dose at a normal dose, the number of molecules required to transport the given dose of a drug is less. So in such a situation, what happens? The transporter molecules are not saturated; they easily transport the drug across, and therefore there is no saturation. If you the same thing, when the same drug is administered at a very high dose, what happens? The large number of drug molecules are available, and the number of transporter molecules will be very small. In such a situation, what happens? The drug transportation, the movement of the drug across the compartments in the body gets affected, and as a result, its pharmacokinetic changes. Therefore, it's important to assess the kinetics of a drug at large doses to understand toxicokinetics. This data will further help us to design how to treat a toxic condition. So somebody consumes this drug in a large doses, and you can know what is the volume of distribution. You can design your treatment options by using this toxicokinetic data, and this is one of the reasons one should generate toxicokinetics data during clinical toxicity studies. And then drug nanocarrier metabolism and excretion data should be available, should be generated, and the occurrence of toxicity is related or correlated with the API or nanocarrier should be established. That means that you need to clearly demonstrate that there is a dose correlation exists, either a dose correlation exists between the API and the occurrence of the toxicity, or a dose correlation exists between the nanocarrier and the occurrence of a toxicity. Otherwise, you should demonstrate that there is no dose correlation exists. This has to be very clearly demonstrated while assessing the animal toxicology data. Obviously, as we all know that nanoparticles are immunogenic in nature, one has to make sure that nanoparticles are immunologically safe by measuring various immunological parameters. So, nanopharmaceuticals clinical safety evaluation. So some of the toxicological studies which are done to assess the preclinical safety of nanoformulations include acute toxicity studies, acute oral, dermal and inhalation toxicity studies, local toxicity studies like acute eye irritation, acute skin irritation, skin sensitization, depending on the route of administration of the nanoformulations. These studies are warranted. And repeated dose systemic toxicity studies are very important part of toxicity studies, which include 14 days repeated dose toxicity studies, 28 days repeated dose toxicity studies, 90 days repeated dose toxicity studies, and 180-day repeated dose toxicity studies. And drugs which are used for more than 6 months normally require carcinogenicity studies and genotoxicity studies. And drugs used in special population may also require reproductive toxicity studies. Normally, our regulators require all these toxicity studies to be conducted in 2 species, preferably one rodent species, another one is a non-rodent species. Rodent species include rat, mice, guinea pigs, and non-rodent species may be, you may have rabbit or you can have dogs. So, preclinical safety data requirement for medications which are intended for a particular duration in clinical. For example, for a orally administered drug, which is used as a single dose or used as a several doses in one day or up to 1 week, normally require 2 weeks of animal toxicology data with 2 different species. A drug which is used for more than 1 week and up to 2 weeks normally requires 4-week toxicology data. So normally the regulation says that normally whenever the duration of usage in humans is say 1 week, you normally do at least 4 times more than that in the, and you expose the drug for 4 times more so as to be safe and also generate meaningful data. I'll just quickly go through these toxicology studies. I will just be giving a brief insight into the acute toxicity because of the want of time, acute toxicity studies and repeated dose toxicity studies, and the general design of study design which is employed to evaluate the safety of these nano formulations, which is same as that which is followed for the regular pharmaceutical formulation. Acute toxicity studies, acute toxicity basically refers to adverse effects which occur within a short period of time, mostly within a period of 14 days after exposure to a single large dose of a chemical or a xenobiotic, or after administration of acute administration of several small doses within a short period of 24 hours. So whatever the adverse effects which you see of this is considered as acute toxicity, and acute toxicity is the first step in any toxicity test assessment. And acute toxicity basically tries to determine the LD50, and acute toxicity data will help us classify the drugs into different categories. Normally, according to the global harmonized system of classification, xenobiotics are classified into category 1, category 2, category 3, category 4, and category 5. If you see a chemical bottle, you will observe this, which category it belongs to. Category 1 is considered as highly hazardous, whose LD50 is normally less than 5 mg per kg body weight, and then category 2 is 50 mg per kg body weight. That means compounds which have LD50 between 5 to 50 are categorized into category 2. Category 3, 300, category 4, 2000, and category 5 is having a LD50 greater than 5000. This category 5 is considered as unclassified, or they are one of the safest categories of xenobiotics. Then coming to the various methods for assessing the acute toxicity, OECD guidelines has 4 methods. Actually, 401 is nowhere, it's removed from the OECD, which is the traditional acute toxicity test, which I'm trying to explain briefly so that you can understand the importance of the other methods. OECD 420 is the fixed dose procedure, OECD 423 is the acute toxic class method, and OECD 425 is up and down procedure. So when compared to OECD 401, OECD 420, 423, and 425 methods can be considered as an alternative methods to these OECD 420, because they all apply the principles of 3 R's, that is the reduction principle of 3 R's, and here the number of animals used will be significantly less when compared to OECD 401. In general, acute toxicity studies are normally done in 2 species. One is a rodent, another one is a non-rodent. Sorry for the typo here. And normally drug may be administered by oral, dermal, or inhalation route. And then parameters measured in acute toxicity studies are clinical signs. Every day you will measure the clinical signs. And then body weight, weekly body weights are recorded, and any deaths which occur during the study period. And also in the end of the study period, normally the study period is for 14 days. And after the 14th day, on 15th day, animals will be cleared to look for the gross necropsy. Here, histopathology will only be done when if there is any gross lesions are observed in the organs. And such organs will give an indication for the next repeated dose toxicity studies, which organs to focus. So in acute toxicity, we determine LD50 or LC. Let us quickly understand the traditional acute toxicity, CD401, how this traditional acute toxicity is done. So in traditional acute toxicity, what we do is normally we divide the animals into 8 to 10 groups. In this example, I have shown 8 groups. Animals were divided into 8 groups, each group containing 10 animals each. And for each group, a different dose of drug is given. For example, in this example, we are given a dose of 10 to 1200 mg per kg weight dose. And then after giving an acute single dose of the drug, animals were observed for clinical signs, mortality, and body weight changes during the study period for a period of 14 days. For example, I have seen the number of deaths in group 1, group 2, there are no deaths, and group 2, group 4, group 6, group 5, 6, and 7 and 10 have different number of animals death, and the percentage mortality is calculated, and also dose is converted to log dose to compress the x-axis, and the log dose versus percentage mortality was plotted, and it is interpolated at 50% to determine the LD50. So this is how LD50 is determined in a traditional method. And if you observe the traditional method of LD50 estimation, determining, although it is accurate determination of the LD50, but it uses a lot of animals. Approximately 80 animals were used in this study, which was really unethical and which can be further improved in the other methods of OECD, that is the OECD 420. And in OECD 420, what they do is it has got 2 steps. First study is the sighting study, in which you use one animal. It is before that OECD 420, there is a fixed dose procedure. Instead of grouping the animals into different groups, here we go step by step. That means you take one animal or 5 animals at a step, and then based on the results, give drug. Based on the results, you decide what dose has to be given to the next set of animals. So here, step-by-step procedure is employed to determine the LD50. If we remember, in fixed dose procedure, we do not accurately determine the LD50. We classify the drug into a HS category 5, 1 to 5. So in case of fixed dose procedure, there are 2 steps. The first step is the sighting study, in which you try to do a study by using one animal at a step to determine what is the appropriate starting dose for the main study. So in this example, you start giving a dosing the first animal at 5 mg per kg. This dose is in turn decided based on the available literature and information on the compound or similar compound toxicity. So you start at 5 mg, and if the animal shows evidence of toxicity, or if the animal dies, then you consider classify the drug directly as a GHS category 1. It is highly toxic. If the animal survives, you give the next higher dose, that is 50 mg per kg. If the animal further survives, you go to the next dose, that is 300 mg per kg. And then if the animal further survives, you go to the 2000 mg per kg. If the animal dies at this stage, then your starting dose will be 300 mg per kg. There's a clear flow diagram which is given to decide what is the starting dose for the main study. Once you decided what is the starting dose, then you carry out the main study by using this starting dose, using 5 animals per step. So you administer 5 animals with the 5 mg per kg. Then you see the results. Based on these results, you decide whether to give a higher dose or a lower dose, or to classify the drug under the GHS category. If the 5 mg per kg, if all the animals die or animals die, more than deaths occur, then you decide directly the LD50 of this drug is, or the toxicity of this drug, LD50 will be around 5 mg per kg. Then further improvisation on this fixed dose procedure is the 423 guideline acute toxic class method. In this method, instead of it does not have a sighting study. Here, the starting dose is decided based on the available information. For example, in this example, here you use, instead of 5 animals, you use 3 animals per step, and which is a much more ethical, where we use 3 animals and still achieve the same degree of quality of results. So here you use the 3, starting with the 300 mg per kg, use 3 animals. More than for 3 animals show mortality or morbidity, then you reduce the dose to 50 mg per kg, and then further there is a toxicity is observed, then you test at 5 mg per kg, and if there is a death, in more than 2 animals, then you classify this as category 0, category 1, that is the toxicity between 0 to 5 mg, that LD50 between 0 to 5 mg. And in case the animals survive, and then you test at 2000 mg per kg, and then if they survive, then you classify the drug as HS category 5. And further 5000 mg testing can also be done. But for that, you require a justification. Normally, beyond 2000 mg limit dose, testing is not permitted because of the ethical reasons, because the animals, stomach is not big enough to consider that. We can, some, formulations such as nutraceuticals may be tested up to 5000 mg per kg. Such type of justification has to be given. The most refined method of OECD method determination of LD50 is the OECD 25, which is also called as up and down procedure. Here you use 1 animal per step instead of 3 animal per step as observed in the acute toxic class method. So here what we do is we follow a dose progression, and a software is also available freely for helping us to decide what dose has to be given next and how to terminate the study. So here we follow a dose progression of 3.2. So every previous dose is multiplied by 3.2 to decide what is the next dose. For example, dose progression of 1.75, 5.5, 17.5, 55, 175, 550, and 2000 are the normal dose progressions which are employed in this toxicity studies. So here you can see in this example, animals were first dose with 175 and 0 indicates survival. They all survived. Next dose is 550 is given, they survived, and at 1750 the next dose, all the animals died. Again you decide come down and give 550, animals survive, and again give 1750, animals die. And it's an error here. Once again you confirm it, and there is a 3 reversals, therefore your study gets completed. And here the LD50, the software estimates as 1750 mg per kg. So unlike OECD 420 and 421 and 423, the OECD 425, like traditional method, gives an accurate estimate of LD50. So this is how LD50 can be determined by using active toxicity studies for a nano-pharmaceuticals. Coming to the repeated dose toxicity studies. So repeated dose toxicity studies are a very important part of any toxicity study estimation. Repeated dose toxicity studies, unlike acute toxicity studies, they will give more information. Repeated dose in acute toxicity, you only get lethal dose 50, nothing much beyond. Many regulators are, for fighting to stop these acute toxicity studies because they don't give much information. But however, acute toxicity data can be used for deciding what is the dose for repeated dose toxicity studies. Also, acute toxicity gross necropsy studies can also give some indication on target organs which are going to be affected by administration of drug. So, to some extent, they only provide minimal information for repeated dose toxicity studies. Repeated dose toxicity, systemic toxicity studies, here drug is repeatedly exposed by oral or by drinking water or via diet, and we are going to assess overall toxicity on the entire system or entire body organs, and this is more comprehensive type of toxicity study. So repeated dose toxicity studies can be subchronic or chronic studies. Subchronic studies are for duration of 28 to 90 days, and above 90 days are considered as chronic studies. So as per the route of administration can be equivalent to the clinical route of administration. So number of animals used can be 5 males and 5 females, and this is a regulatory requirement. However, toxicological requirement can be more. If you use more animals, it is more appropriate for statistical analysis. But however, regulatory requirement is minimum of 5 animals and 5 males and 5 females should be used for each dose level. Only the study design of this repeated dose toxicity, either it could be 14 days, 28 days, 90 days, or 180 days. The study design remains the same. So you will have a control group in which vehicle will be administered. You will have a low dose group, high dose group, a mid dose group. Only this is the common study design. In additionally or optionally, you can have a satellite group observe reversibility of the toxicity or delayed occurrence of the toxic effects. Okay. So this is satellite group. Normally what happens if the toxicity study is for 28 days, in addition to 28 days, at the end of the 28 days, all the 4 groups will be sacrificed, and they are sacrificed, and their the all the parameters were analyzed, whereas the fifth group is left like that without treatment for another 14 days to look for the reversibility of the toxicity and also for other additional measurements. Observations include clinical signs, mortality, such as health condition, changes in the skin, fur, eye, and all those things. And weekly body weight is recorded to indirectly measure the effect of drug on the body weight and also health of the animals. Feed intake will also give you an important indication. This is one of the table I just wanted to show how the data is recorded. Clinical signs are recorded in a 28-day repeated dose toxicity studies. And this is an example for how weekly body weight is recorded and analyzed. And this is an example of how food intake is recorded and analyzed. And further, at the end of the study, before the sacrifice, at 28 day, normally, clinical laboratory investigation is done by collecting the blood, like hematological parameters, clotting time, and blood smears were prepared for differential count. And also the blood is also used for biochemical parameter estimation, like fasting glucose, total cholesterol, and all these parameters were analyzed to identify the possible toxic effects on various organs. For example, this parameters include liver function test, kidney function test, cardiac function test. So many tests are there to identify the possible toxicity on various organs. Further, on the 4th week before blood collection, what we do is we also collect the urine to analyze the urine analysis to identify possible toxicity on the urine. After blood collection and urine collection, at the end of the study, animals were sacrificed by using anesthetic, excess dose of anesthesia, and all organs were collected, and some of the organ weights are normally measured, such as brain, heart, spleen, thymus, gonads, adrenals, kidney, and all these organs were measured. Their weight is taken, and then they were subjected to histopathological analysis. In addition to all other organs were collected, except even the skin, except the skin and the tail. All other organs from the animals were collected and subjected to a detailed histopathological analysis. So these are some of the tables how hematological data is collected and analyzed. This is a table for biochemical analysis. This is the urine analysis, and this is the table which shows how the organ weights were analyzed, and this is the analysis of the histopathology, how histopathology data is analyzed. And finally, the whole purpose of this repeated dose toxicity studies is to determine no observed adverse effect level, NOAEL. Application of this NOAEL is to determine the reference dose for clinical trials. Reference dose is calculated based on the NOAEL, which is being determined in the repeated dose toxicity study. So how do you decide what is the NOAEL? For example, this study I have used, a control group, a low dose group, high dose group, and the mid dose group and the high dose group. So low dose group is 100, mid dose group is 500, and the high dose group is 1000. So I will analyze this data and I will observe that 1000 mg per kg, there is no toxicity observed, significant difference in the toxicity was observed between the control group and the high dose group. Then I can consider the NOAEL for this drug as 1000 mg per kg. Now this 1000 value is used in the reference dose calculation formula, which is divided by normally by 100 factor. So this 10 factor is accounted for uncertainty factor, and 10 is for the modifying factor. So reference dose determination, therefore, repeated dose toxicity data will help us to determine the first dose or the reference dose for the phase 1 clinical trials. So with this, I would like to conclude. I could have given many other things, but I don't think that is, I think, beyond the scope of this presentation. So in conclusion, I would like to say nanotherapeutics offer many advantages over conventional therapeutics, especially in the treatment of diseases like cancer. The current challenges in the development of nanotherapeutics is optimizing their physicochemical properties, doing large-scale manufacturing, and efficient assessment of their efficacy and safety. So with this, I'd like to thank you all for your participation in this webinar. And also I take this opportunity to thank organizers for giving me an opportunity to just share my knowledge with regard to nano-pharmaceuticals and their safety assessment. Thank you. If you have any questions, I'm happy to take.
Okay, thank you, sir. Now the session is open for questions. Anyone in the Zoom live can directly post the questions to the guest speaker. Those in the chat box from the YouTube live, I will convey those questions to the sir. Anybody open for questions? Sir, Sir, here is a question from YouTube chat box, sir. This is from Dr. K. Ramireddy, Associate Professor from University College of Pharmaceutical Sciences, ANU. What is the most feasible OECD guideline for accessing acute toxicity of nanoparticles?
As I told you, OECD has got many guidelines. So, and see, any guideline can be used, for example, for what is the advantage of different guidelines? For example, if you want to determine the accurate LD50 estimation, it would be appropriate to go by the traditional method. Traditional method you cannot use now. It's unethical, no ethical committee will approve. The best method would be OECD 425, because OECD 425 will give you accurate estimate of the LD50. Because nanoparticles are highly relatively toxic, could we have some toxicity? Therefore, it would be appropriate for us to have accurate determination. Whereas other methods like OECD 420 and 423 will only give you a range estimate of the LD50, will not give you an accurate estimate of the LD50. I would suggest to go with the OECD 425 guidelines, up and down procedure.
Thank you, sir. And here is one more question from Dr. S.K. Arifa Begum, Associate Professor from Chelapathy Institute of Pharmaceutical Sciences, Guntur. And the question is, how do we select dose after acute toxicity studies for nanomaterials? Okay,
so for dose for what studies? I did not get for what studies. Is it for repeated dose toxicity studies or efficacy studies? Okay, whatever. I'll explain for both.
Yes.
See, acute toxicity data will tell you about the LD50, lethal dose. Normally, what we do is for efficacy studies, we go 1/10 below the LD50. That means if your LD50 is 2000 mg, you divide that value by 10 times. So you get 200 mg per kg. Normally, the highest dose in efficacy studies, we use 200 mg per kg, but there is no hard and fast rule in this. But normally, this is how we decide. And also, that you need to decide this further, you need to take into consideration the related substance toxicity. For example, you have a similar structures which are already existing whose LD50 is known. You also take into consideration while deciding these factors. In general, what they do is normally they divide the LD50 value with 1/10. This dose will be taken as the highest dose for efficacy studies. And even for repeated dose toxicity studies, normally this dose is taken as the highest dose for the repeated dose toxicity studies as well. But this is not a very accurate rule. It's a general rule accepted now.
Okay, thank you, sir. And further, anyone who are inhibiting in asking the questions online or are live, we are, with your permission, we would like to share your email ID so that they can approach you with their questions.
No problem, sir.
Okay, thank you very much, sir. And I request our principal, sir, Dr. K. Prasad Garu, to give the concluding note for today's session as a complete FDP program.
Thank you, Dr. Praveen.
Thank you, sir.
Excellent lectures combined with most of the nanoparticles related to pharmacology in depth, how to do the pharmacological experiment by using this one. So today's lecture is all parts are combined, starting from the Sutics to pharmacology, and thank you for this very interesting lecture and most useful for the pharmacology who are doing these activities on by using the nanoparticles. Thank you.
Yes, sir.
Regarding this 5 days program, so in during these 5 days, 6 days programs we are having, sir, from the on behalf of the Sri Vishnu Educational Society as well as Devraj Foundation and Vishnu College of Pharmacy. I thank all the speakers who are eminent professors from different colleges, Dr. Ramanamurthy from Andhra University, Dr. G. Chandraya from Naipur, Hyderabad, Dr. Harnath, NIT Warangal, and Dr. P. Veerabrahmachari, Krishna University, and then Dr. Professor Abhimanyu from SRM University, and then Professor Praveen from Uti, Day-6 College of Pharmacy, Uti. They have given excellent information regarding the nanotechnology. All the details have been covered during these 5 days regarding the starting from the preparation of the nanoparticles, and then today it has ended how to do the test by using the nanoparticles in pharmacological studies. So I am very much thankful for all these speakers on behalf of Sri Vishnu Educational Society. So if anything is related to this information or any data is required, we will be in touch with you in the process of doing the research programs. Once again, I thank all the speakers and then participants. Actually, we are having more than 1150 participant delegates from the different colleges from throughout India. I thank all the participants, and then I request the same participation in the coming next week. We are conducting another webinar. So I request all these participants to register for these coming programs and cooperate with us. Thank you.
Over to Ashok.
Over to Ashok for the.
Okay, thank you, sir. Thank you very much. Now it is my privilege to convey my vote of thanks. On behalf of Sri Vishnu College of Pharmacy, Bhimavaram, I would like to propose my heartfelt gratitude and vote of thanks to Sri K. Vishnu Raju Garu, Chairman, Sri Ravichandran Rajagopal, Vice Chairman, Sri Vishnu Educational Society and Dr. B.V. Raju Foundation, Dr. Kumar V.S. Nemani, Director, Dr. K. Prasad, Principal, Sri Vishnu College of Pharmacy, Bhimavaram, Dr. S. Vidyadhara, Chairman of IPA Education Division, Dr. Ramchandra Rao K. Director for Crystal Growth and Nanoscience Research Center, Government College, Autonomous, Rajahmundry, for associating with us in conducting this FDP on nanotechnology in healthcare. Similarly, I would like to convey my gratitude to the honorable guest speakers, Professor Ramanmurthy Garu from Andhra University, Principal Andhra University College of Pharmaceutical Sciences, Dr. G. Chandraya from NIPER Hyderabad, Dr. Harnath from NIT Warangal, Dr. P. Veera Brahmachari from Krishna University, Professor Abhimanyu from SRM College of Pharmacy, Professor Praveen T.K., Professor and HOD of Pharmacology from JSS Ooty, for being with us and sharing their valuable knowledge. Apart from that, I would like to also thanking our teaching and technical staff. Also, I would like to propose my special thanks to Dr. A. Ramesh, Principal, Viper, our sister institution, Dr. C.H.S. Panikumar, Principal, Adarsha College of Pharmacy, Kottapalli, Dr. Mohan Gandhi, Associate Professor from Vasavi College of Pharmacy, Tadepallipuram, Dr. K. Nageshwar Rao Garu, Director, KGRL Institutions, Bhimavaram, for being a constant support to us and constant motivation to us to conduct these sort of activities. Thank you, sir. Now I declare the session has been closed. One more request to all the participants for making this event a grand success. Lastly, your feedbacks, you are requested to submit your feedbacks, which is highly appreciated to upgrade ourselves. Thank you to one and all. Now I declare the session as well as the FTP or nanotechnology in healthcare is closed. Thank you to one and all.